The basic component in the nervous system is the nerve cell or neuron, composed of a cell body with two projections fibers, the dendrites that receive stimuli and the axon that transmits information, either to another neuron or to an effector organ such as a muscle. Axon may have lateral branches called collateral and terminal arborization and synapse. Insect neurons release a variety of chemicals at synapses either to stimulate or to inhibit effector neurons or muscles. Acetylcholine and catecholamines such as dopamine are the important neurotransmitters involved in the impulse conduction. Neurons are of following types based on structure and function. A. On structural basis. I. Monopolar, neuron with a single axon. E. Bipolar, neuron with a proximal axon and a long distal dendrite. E. Multipolar, neuron with a proximal axon and many distal dendrites. B. Functional basis. I. Sensory neuron, it conducts impulse from sense organs to central nervous system. CNS. E. Motor neuron, it conducts impulse from CNS to effector organs. E. Interneuron, association neuron, it interlinks sensory and motor neurons. The cell bodies of interneurons and motor neurons are aggregated with the fibers interconnecting all types of nerve cells to form nerve centers called ganglia. Mechanism of impulse conduction. Impulses are conducted by the neurons by two means. Axonic conduction. Ionic composition varies between inside and outside of axon resulting in excitable conditions, which leads to impulse conduction as electrical response. Synaptic conduction. Neurochemical transmitters are involved in the impulse conduction through the synaptic gap. Neurotransmitters and the type of reactions helping in the impulse conduction are as follows. Nervous system can be divided into three major subsystems as I. Central nervous system, CNS. E. Visceral nervous system, VNS. E. Peripheral nervous system, PNS. I. Central nervous system. It contains double series of nerve centers, ganglia. These ganglia are connected by longitudinal tracts of nerve fibers called connectives and transverse tracts of nerve fibers called commissures. Central nervous system includes the following. A. Brain, formed by the fusion of first three cephalic neuromers. Protocerebrum, large, a nervate compound eyes and ocelli. Dedocerebrum, found beneath protocerebrum, a nervate antennae. Tritocerebrum, bilobed, a nervate labrum. Brain is the main sensory center controlling insect behavior. B. Ventral nerve cord, median chain of segmental ganglia beneath esophagus. C. Subesophageal ganglia, formed by the last three cephalic neuromers which innervate mandible, maxillae and labium. D. Thoracic ganglia, three pairs found in the respective thoracic segments, largest ganglia, innervate legs and muscles. E. Abdominal ganglia, maximum 8 pairs will present and number varies due to fusion of ganglia. Innervate spiracles. F. Thoraco-abdominal ganglia, thoracic and abdominal ganglia are fused to form a single compound ganglia. Innervate genital organs and cerci. E. Visceral nervous system. The visceral sympathetic nervous system consists of three separate systems as follows. 1. The stomatogastric, which includes the frontal ganglion and associated with the brain, aorta and foregut. 2. Ventral visceral, associated with the ventral nerve cord. And 3. Caudal visceral, associated with the posterior segments of abdomen. Together the nerves and acetylase. Acetyl Ganglia of these subsystems innervate the anterior and posterior gut, several endocrine organs, corpora cardiaca and corpora allata, the reproductive organs, and the tracheal system including the spiracles. E. Peripheral nervous system. The peripheral nervous system consists of all the motor neuron axons that radiate to the muscles from the ganglia of the CNS and visceral nervous system plus the sensory neurons of the cuticular sensory structures, the sense organs, that receive mechanical, chemical, thermal or visual stimuli from an environment. Lecture 13. Anatomy. Reproductive system. Reproductive system. It is important to note here that variation among insect reproductive systems is great. Closely related species are often isolated from one another via small variations in the morphology of reproductive organs that prohibit interspecies mating. However, a generalized system can be constructed that closely represents all sexually reproducing insects. Do not be alarmed if you are unable to locate the indicated structures on your dissected specimens. Be familiar with differences in male and female genitalia and be able to identify structures. Do not be alarmed if you are unable to locate the indicated structures on your dissected specimens. Be familiar with differences in male and female genitalia and be able to identify structures when given a diagram. Directions are provided if you wish to attempt seeing the reproductive system of your specimen. The female reproductive system. Remove the digestive tract from your female specimen by cutting it with a scalpel at the posterior and anterior most regions. Place digestive tract to the side. If necessary, remove muscle tissue, as well, until the ovaries, lateral oviducts, median oviduct, seminal receptacle and vagina are visible. Compare your specimen with the figures below. Then observe classmates' specimens. The male reproductive system. Remove the digestive tract from your male specimen by cutting it with a scalpel at the posterior and anterior most regions. Place digestive tract to the side. If necessary, remove muscle tissue, as well, until the testes, vas deferens, seminal vesicle, accessory glands, and ejaculatory duct are visible. Compare your specimen with the figures below. Then observe classmates' specimens. In insects, male and female sexes are mostly separate. Sexual dimorphism is common where the male differ from the female morphologically as in bees, mosquito and cockroach. Sexual dimorphism is common where the male differ from the female morphologically as in bees, mosquito and cockroach. The other types are gynandromorph, sexual mosaic, abnormal individual with secondary sexual characters of both male and female, e.g. mutant Drosophila. Hermaphrodite, male and female gonads are present in one organism, e.g. cottony cushion scale. Female reproductive system. The main functions of the female reproductive system are egg production and storage of male spermatozoa until the eggs are ready to be fertilized. The basic components of the female system are paired ovaries, which empty their mature oocytes, eggs, via the calyx's calyx, into the lateral oviduct which unite to form the common, median, oviduct. The gonopore opening of the common oviduct is usually concealed in an inflection of the body wall that typically forms a cavity, the genital chamber. This chamber serves as a copulatory pouch during mating and thus is often known as the bursa copulatrix. Its external opening is the vulva. In many insects the vulva is narrow and the genital chamber becomes an enclosed pouch or tube referred to as the vagina. Two types of ectodermal glands open into the genital chamber. The first is the spermatheca which stores spermatozoa until they are needed for egg fertilization. The spermatheca is single and sac-like with a slender duct, and often has a diverticulum that forms a tubular spermathecal gland. The gland or glandular cells within the storage part of the spermatheca provide nourishment to the contained spermatozoa. The second type of ectodermal gland, known collectively as accessory glands, opens more posteriorly in the genital chamber. Each ovary is composed of a cluster of egg or ovarian tubes, the ovarioles, each consisting of a terminal filament, a germarium in which mitosis gives rise to primary oocytes, a vitellarium in which oocytes grow by deposition of yolk in a process known as vitellogenesis, and a pedicel. An ovariole contain a series of developing oocytes each surrounded by a layer of follicle cells forming an epithelium. The oocyte with its epithelium is termed a follicle. The youngest oocyte occur near the apical germarium and the most mature near the pedicel. There are different types of ovarioles based on the presence or absence of specialized nutritive cells called trophocytes, nurse cells for nourishment of oocytes. Poneostic ovariole lacks specialized nutritive cells so that it contains only a string of follicles, with the oocytes obtaining nutrients from the hemolymph via the follicular epithelium. E.g. cockroach. Telotrophic ovariole. Acrotrophic, the trophocyte is present and its location is confined to the germarium and remain connected to the oocytes by cytoplasmic strands as the oocytes move down the ovariole. E.g. bugs. Polytrophic ovariole. A number of trophocytes are connected to each oocyte and trophocytes moves down along with the ovariole, providing nutrients until depleted. Thus individual oocytes are alternated with groups of smaller trophocytes in the ovarioles. E.g. moths and flies. Accessory glands of the female reproductive tract are often called as collateral or cement glands, because their secretions surround and protect the eggs or cement them to the substrate. E.g. egg case production in mantis, ootheca formation in cockroach, venom production in bees. Male reproductive system. The main functions of the male reproductive system are the production and storage of spermatozoa and their transport in a viable state to the reproductive tract of the female. Morphologically, the male tract consists of paired testes, each containing a series of testicular tubes or follicles, in which spermatozoa are produced, which open separately into the sperm duct or vas deferens. This vas deferens expands posteriorly to form a sperm storage organ or seminal vesicle. Tubular paired accessory glands are formed as diverticula of the vasa deferentia. Sometimes the vasa deferentia themselves are glandular and fulfill the functions of accessory glands. The paired vasa deferentia unite where they lead into ejaculatory duct, the tube that transports the semen or the sperm to the gonopore. Accessory glands are 1 to 3 pairs associated with vasa deferentia or ejaculatory duct. Its function is to produce seminal fluid and spermatophores, sperm-containing capsule. Physiology of reproduction. Spermatogenesis. Occurs inside sperm tube. Oogenesis. Occurs inside egg tube. Spermatogonia oogonia. Mitosis. Primary spermatocytes 2N, primary oocytes 2N, meiosis. Secondary spermatocytes N, secondary oocytes N, mitosis. Spermatids N, oocytes N, spermiogenesis, vitellogenesis, sperms N, ovum N. 3. Sperm transfer. I. Intragenital. Common method. Through edagous via vaginal orifice into female genital passage. E. Hemocoelus. Sperms transferred into the body cavity e.g. bed bug. E. External. Spermatophores are ejected out into open place by male while female walk over it and gets inseminated e.g. silverfish. 4. Fertilization. Sperm enter into egg to produce morphogenesis. Egg nucleus divides meiotically into female gamete nucleus and polar body. Then the fertilization occurs by the fusion of male and female gamete nuclei. Types of reproduction. 1. Oviparity. Majority of female insects, or oviparous, lay eggs. Embryonic development occurs after oviposition by utilizing the yolk, e.g. headlouse, moths. 2. Viviparity. Unlike oviparous, here initiation of egg development takes place within the mother. The life cycle is shortened by retention of eggs and even developing young within the mother. 4 main types of viviparity are observed in different insect groups. I. Ovoviviparity. Fertilized eggs containing yolk are incubated inside the reproductive tract of the female and hatching of egg occur just prior to or soon after oviposition e.g. thrips, some cockroaches, few beetles, and flesh fly. Fecundity of this group is low. E. Pseudoplacental viviparity. This occurs when a yolk-deficient egg develops in the genital tract of the female. The mother provides a special placenta-like tissue, through which nutrients are transferred to developing embryos. There is no oral feeding and larvae are laid upon hatching, e.g. aphids, some earwigs, sosids and polytenid bugs. E. Hemocoelus viviparity. This involves embryos developing free in the female's hemolymph with nutrients taken up by osmosis. This form of internal parasitism occurs only in Strepsiptera and some gall midges. IV. Adenotrophic viviparity. This occurs when a poorly developed larva hatches and feeds orally from accessory gland, milk gland secretion within the uterus of the mother. The full-grown larva is deposited and pupates immediately, e.g. tsetse flies, louse, ked, bat flies. 3. Parthenogenesis. Reproduction without fertilization is parthenogenesis. Different types of parthenogenesis are as follows. A. Based on occurrence. I. Facultative, not compulsory, e.g. B. E. Obligatory or constant compulsory, e.g. stick insect. E. Cyclic, sporadic, alternation of gamic and agamic population, e.g. aphid. B. Based on sex produced. I. Urinotoki, produce male, e.g. bee. Thelitoki, produce female, e.g. aphids. E. Amphitoki, deuterotoki, produce both male and female, e.g. cynipid wasp. C. Based on meiosis. I. Apomictic, no meiosis occurs. E. Automictic, meiosis occurs, but diploidy is maintained. 4. Polyembryony. This form of asexual reproduction involves the production of 2 or more embryos from one egg by subdivision. Mostly observed in parasitic insects, e.g. Platygaster. Nutrition for a large number of developing embryo cannot be supplied by the original egg and is acquired from the host's hemolymph through a specialized enveloping membrane called trifamnion. 5. Paidogenesis. Some insects cut short their life cycles by loss of adult and pupal stages. In this precocious stage, gonads develop and give birth to young one by parthenogenesis, i.e. reproduction by immature insects. I. Larval paidogenesis, e.g. gall midges, e. Pupal paidogenesis, e.g. Meister, sp. Sense organs. Sensilla are the organs associated with sensory perception and develop from epidermal cells. The different types of sense organs are 1. Mechanoreceptors. 2. Auditory receptors. 3. Chemoreceptors. 4. Thermoreceptors and 5. Photoreceptors. 1. Mechanoreceptors detect mechanical forces. I. Trichoid sensilla, hair-like little sense organ. Sense cell associated with spur and seta. These cells are sensitive to touch and are located in antenna and traphe, mouth parts. E. Campaniform sensilla, dome sensilla, terminal end of these sensilla is rod-like and inserted into dome-shaped cuticula. These cells are sensitive to pressure and located in leg joints and wing bases. E. Chordotonal organ. The specialized sensory organs that receive vibrations are subcuticular mechanoreceptors called chordotonal organ. An organ consists of one to many scolopidia, each of which consists of cap cell, scallopale cell and dendrite. These organs are interoceptors attached to both ends of body wall. Functions. I. Proprioception, positioning of their body parts in relation to the gravity. E. Sensitive to sound waves, vibration of substratum and pressure changes. E. Johnston's organ. All adults, insects and many larvae have a complex chordotonal organ called Johnston's organ lying within the second antennal segment pedicel. These organs sense movements of antennal flagellum. It also functions in hearing in some insects like male mosquitoes and midges. IV. Subgenual organ. Chordotonal organ located in the proximal tibia of each leg, used to detect substrate vibration. Subgenual organs are found in most insects, except the Coleoptera and Diptera too. Auditory receptors detect sound waves. I. Delicate tactile hairs, present in plumose antenna of male mosquito. E. Tympanum. This is a membrane stretched across tympanic cavity, responds to sounds produced at some distance, transmitted by airborne vibration. Tympanal membranes are linked to chordotonal organs that enhance sound reception. Tympanal organs are located between the metathoracic legs of mantids. The metathorax of many nectoid moths. The prothoracic legs of many orthopterans. The abdomen of short-horned grasshopper, cicada. The wings of certain moths and lacewings. 3. Chemoreceptors detect smell and taste. Detect chemical energy. Insect chemoreceptors are sensilla with one pore, uniporous, or more pores, multiporous. Uniporous chemoreceptors mostly detect chemicals of solid and liquid form by contact and are called as gustatory receptor. Many sensor neurons located in antenna are of this type. Multiporous chemoreceptors detect chemicals in vapor form, at distant by smell and are called as olfactory receptor. Few sensory neurons located in traphe and tarsi are of this type. Each pore forms a chamber known as pore kettle with more number of pore tubules that run inwards to meet multibranch dendrites. 4. Thermoreceptors detect heat. Present in poikilothermic insects and sensitive to temperature changes. In bed bug it is useful to locate the host utilizing the temperature gradient of the host. 5. Photoreceptors detect light energy. A. Compound eyes. The compound eye is based on many individual units called ommatidia. Each ommatidium is marked externally by a hexagonal area called facet. Compound eye is made up of 2 parts called optic part and sensory part. Optic part contains a cuticular lens called corneal lens secreted by corneginous cells and crystalline cone covered by primary pigment cells. Function of the optic part is to gather light. Sensory part contains 6 to 10 visual cells called retinular cells covered by secondary pigment cells which collectively secrete a light-sensitive rod at the center called rhabdom. Rhabdom contains light-sensitive pigments called rhodopsin. Each ommatidium is covered by a ring of light-absorbing pigmented cells, which isolates an ommatidium from other. Nerve cells are clustered around the longitudinal axis of each ommatidium. Types of ommatidia. I. Apposition type, light tight. Due to the presence of primary pigment cells, light cannot enter the adjacent cells. The mosaic image formed is very distinct. The image formed by the compound eye is of a series of opposed points of light of different intensities. This functions well in diurnal insects. E. Superposition type. Primary pigment cells are absent, allowing light to pass between adjacent ommatidia. Image formed in this way are indistinct, bright and blurred. This type is seen in nocturnal and crepuscular insects. B. Lateral ocelli, stemata, visual organs of holometabolous larva. Structure is similar to Ommatidium. It helps to detect form, color and movement, and also to scan the environment. C. Dorsal ocelli, visual organs of nymph, and it vary from 0 to 3 in numbers. It contains a single corneal lens with many visual cells individually secreting the rhabdomere. Dorsal ocelli perceive light to maintain diurnal rhythm and is not involved in image perception. Lecture 14. Sensory organs. Metamorphosis. Egg structure and types of eggs. Metamorphosis and immature stages in insects. Metamorphosis is the change in growth and development an insect undergoes during its life cycle from birth to maturity. There are 4 basic types of metamorphosis in insects. 1. Ametabola. No metamorphosis, e.g. silverfish. These insects have only 3 stages in their life, namely egg, young ones and adult. It is most primitive type of metamorphosis. The hatching insect resembles the adult in all respects except for the size and called as juveniles. Molting continues throughout the life. 2. Hemimetabola. Incomplete metamorphosis, e.g. dragonfly, damselfly and mayfly. These insects also have 3 stages in their life, namely egg, young one and adult. The young ones are aquatic and are called as naiads. They are different from adults in habit and habitat. They breathe by means of tracheal gills. In dragonfly naiad, the lower lip, labium, is called mask, which is hinged and provided with hooks for capturing prey. After final molt, the insects have fully developed wings suited for aerial life. 3. Parometabola. Gradual metamorphosis, e.g. cockroach, grasshopper, bugs. The young ones are called nymphs. They are terrestrial and resemble the adults in general body form except the wings and external genitalia. Their compound eyes and mouth parts are similar to that of adults. Both nymphs and adults share the same habitat. Wing buds externally appear in later instars. The genitalia development is gradual. Later instar nymphs closely resemble the adult with successive molts. 4. Holometabola. Complete metamorphosis, e.g. butterfly, moth, fly and bees. These insects have 4 life stages, namely egg, larva, pupa and adult. Majority of insects undergo complete metamorphosis. Larvae of butterflies are called caterpillar. Larva differs greatly in form from adult. Compound eyes are absent in larva. Lateral ocelli or stemata are the visual organs. Their mouth parts and food habit differ from adults. Wing development is internal. When the larval growth is completed, it transforms into pupa. During the non-feeding pupal stage, the larval tissues disintegrate and adult organs are built up. 1. Eggs. The first stage of development in all insects is egg. Majority of insects are oviparous. Egg stage is inconspicuous, inexpensive and inactive. Yolk contained in the egg supports the embryonic development. Eggs are laid under conditions where the food is available for feeding of the future youngones. Eggs are laid either individually or in groups. The outer protective shell of the egg is called chorion. Near the anterior end of the egg, there is a small opening called micropyle which allows the sperm entry for fertilization. Chorion may have a variety of textures. Size and shape of the insect eggs vary widely. Types of eggs. A. Singly laid. 1. Sculptured egg. Chorion with reticulate markings and ridges, e.g. castor butterfly. 2. Elongate egg. Eggs are cigar-shaped, e.g. sorghum shoot fly. 3. Rounded egg. Eggs are either spherical or globular, e.g. citrus butterfly. 4. Knit. Egg of head louse is called knit. It is cemented to the base of the hair. There is an egg stigma at the posterior end, which assists in attachment. At the anterior end, there is an oval lid which is lifted at time of hatching. 5. Egg with float. Egg is boat-shaped with a conspicuous float on either side. The lateral sides are expanded. The expansions serve as floats, e.g. Anopheles mosquito. B. Eggs laid in groups. 1. Pedicellate eggs. Eggs are laid in silken stalks of about 1.25 mm length in one groups on plants, e.g. green lacewing fly. 2. Barrel-shaped eggs. Eggs are barrel-shaped. They look like miniature batteries. They are deposited in compactly arranged masses, e.g. stink bug. 3. Ootheca place. Ootheca eggs are deposited by cockroach in a brown bean-like chitinous capsule. Each ootheca consists of a double-layered wrapper protecting 2 parallel rows of eggs. Each ootheca has 16 eggs arranged in 2 rows. Ootheca are carried for several days protruding from the abdomen of female prior to oviposition in a secluded spot. Along the top, there is a crest which has small pores which permit gaseous exchange without undue water loss. Chitinous egg case is produced out of the secretions of collateral glands. 4. Egg pod. Grasshoppers secrete a frothy material that encases an egg mass which is deposited in the ground. The egg mass lacks a definite covering. On the top of the egg, the frothy substance hardens to form a plug which prevents the drying of eggs. 5. Egg cass. Mantids deposit their eggs on twigs in a foamy secretion called spumoline which eventually hardens to produce an egg case or ootheca. Inside the egg case, eggs are aligned in rows inside the egg chambers. 6. Egg mass. Moths lay eggs in groups in a mass of its body hairs. Anal tuft of hairs found at the end of the abdomen is mainly used for this purpose, e.g. rice stem borer. Female silkworm moth under captivity lays eggs on egg card. Each egg mass is called a DFL, diseases-free laying. 7. F. raft. In Culex mosquitoes, the eggs are laid in a compact mass consisting of 200 to 300 eggs called egg raft in water. Lecture 15. Metamorphosis. Larva and types of larva. Larva. Larval stage is the active growing stage. It is the immature stage between the egg and pupal stage of an insect having complete metamorphosis. This stage differs radically from the adult. Types of larva. There are 3 main types of insect larvae, namely oligopod, polypod and opodus. 1. Oligopod. Thoracic legs are well developed. Abdominal legs are absent. There are two subtypes. A. Campodiform. They are so called from their resemblance to the dipleran genus Campodia. Body is elongate, depressed dorsoventrally and well sclerotized. Head is prognathous. Thoracic legs are long. A pair of abdominal cerci or caudal processes is usually present. Larvae are generally predators and are very active, e.g. grub of antlion or grub of lady brid beetle. B. Scarabaeiform. Body is C-shaped, stout and subcylindrical. Head is well developed. Thoracic legs are short. Caudal processes are absent. Larva is sluggish, burrowing into wood or soil, e.g. grub of rhinoceros beetle. 2. Polypod or Eruciform. The body consists of an elongate trunk with large sclerotized head capsule. Head bears a pair of powerful mandibles which tear up vegetation. Two groups of single-lensed eyes, stemata, found on either side of the head constitute the visual organs. The antenna is short. 3 pairs of thoracic legs and up to 5 pairs of unjointed abdominal legs or prolegs are present. Thoracic legs are segmented and they end in claws which are used for holding onto the leaf. Bottom of the proleg is called planta, which typically bears rows or circlet of short hooked spines or crochets which are useful in clinging to the exposed surface of vegetation and walking. Abdominal segments 3 to 6 and 10 typically bear prolegs, e.g. caterpillar, larva of moths add butterflies. A. Hairy caterpillar. The body hairs may be dense, sparse or arranged in tufts. Hairs may cause irritation when touched, e.g. red hairy caterpillar. B. Slug caterpillar. Larva is thick, short, stout and fleshy. Laval head is small and retractile. Thoracic legs are minute. Abdominal legs are absent. Abdominal segmentation is indistinct. Larva has poisonous spines called scoli distributed all over the body. Such larva is also called platyform larva. C. Semilooper. Either 3 or 4 pairs of prolegs are present. Prolegs are either wanting or rudimentary in either third and fourth abdominal segments, e.g. castor semilooper. D. Looper. They are also called measuring worm or earth measurer or inchworm. In this type, only 2 pairs of prolegs are present in 6th and 10th abdominal segments, e.g. daintia looper. 3. Opodus. They are larvae without appendages for locomotion. Based on the degree of development and sclerotization of head capsule, there are 3 subtypes. A. Euspalus. Larva with well-developed head capsule with functional mandibles, maxillae, stemata and antennae. Mandibles act transversely, e.g. wriggler, larva of mosquito, and grub of red palm weevil. B. Hemicephalus. Head capsule is reduced and can be withdrawn into thorax. Mandibles act vertically, e.g. larva of horsefly and robber fly. C. Acephalus. Head capsule is absent. Mouthparts consist of a pair of protrusible curved mouth hooks and associated internal sclerites. They are also called vermiform larvae, e.g. maggot, larva of housefly. Lecture 16. Metamorphosis, pupa and types of pupae. Pupa. It is the resting and inactive stage in all holometabolous insects. During this stage, the insect is incapable of feeding and is quiescent. During the transitional stage, the larval characters are destroyed and new adult characters are created. There are 3 main types of pupae. 1. Obtect. Various appendages of the pupa viz. Antennae, legs and wing pads are glued to the body by a secretion produced during the last larval molt. Exposed surfaces of the appendages are more heavily sclerotized than those adjacent to body, e.g. moth pupa. A. Chrysalis. It is the naked obtect pupa of butterfly. It is angular and attractively colored. The pupa is attached to the substratum by hooks present at the terminal end of the abdomen called cremaster. The middle part of the chrysalis is attached to the substratum by 2 strong silken threads called griddle. B. Tumbler. Pupa of mosquito is called tumbler. It is an obtect type of pupa. It is comma-shaped with rudimentary appendages. Breathing trumpets are present in the cephalic end and anal paddles are present at the end of the abdomen. Abdomen is capable of jerky movements which are produced by the anal paddles. The pupa is very active. 2. Exarate. Various appendages viz. Antennae, legs and wing pads are not glued to the body. They are free. All oligopod larvae will turn into exarate pupae. The pupa is soft and pale, e.g. pupa of rhinoceros beetle. 3. Coarctate. The pupal case is barrel-shaped, smooth with no apparent appendages. The last larval skin is changed into case containing the exarate pupa. The hard and dark brown pupal case is called puparium, e.g. fly pupa. Pupal protection. In general, pupal stage lacks mobility. Hence it is the most vulnerable stage. To get protection against adverse conditions and natural enemies, the pupa is enclosed in a protective cover called cocoon. Based on the nature and materials used for preparation of cocoons, there are several types. Types of cocoon materials used: example silken cocoon, silk, silkworm, earthen cocoon, soil plus saliva, gram, pod borer, hairy cocoon, body hairs, woolly bear. Frassy cocoon, frass plus saliva, coconut, black-headed caterpillar. Fibrous cocoon, fibers, red clam, weevil, puparium, hardened, last larval skin, house fly. Lecture 17. Taxonomy, principles and procedures. Nomenclature and identification. Taxonomy is the science of classification. It can be defined as placing biological organisms or forms in order. Simpson, 1961, has defined taxonomy as the theoretical study of classification including basis, principles, procedures and rules. Taxonomy includes nomenclature and classification. Systematics, the science of study of kind and diversity of organisms and any or all relations among them. Systematics includes taxonomy and evolution. Stages of taxonomy. The taxonomy of any group passes through several stages. Alpha taxonomy, alpha, it is concerned with naming and characterization of species. Beta taxonomy, beta, concerned with classification. Gamma taxonomy, gamma, concerned with evolutionary relations and phylogeny. Basis for classification. Classification is the ordering of a large group of organisms based on certain characters into small groups. Classification is mainly based on evolutionary relationship and not based on superficial resemblance. Points considered while classifying are I. External structure, E. Internal characters, E. Developmental history, IV. Physiological data and V. Cytogenetic data. The biological system of classification is called hierarchical concept of classification. This was introduced by Carl von Linnaeus, 1758. A large group of organism is successively subdivided into small group. These groups are called taxa, taxon singular. Each group is at a particular level in this system. This level is called the rank. Groups of the same rank are grouped together and that constitutes the taxonomic category. E.g., class. Certain taxonomic categories are obligate, while others are optional. For describing and classifying any organism, the basic taxonomic category is species. The lowermost category for classifying an organism is subspecies. Systematic position of Indian honeybee. 1. Kingdom, Animalia. 2. Phylum, Arthropoda. 3. Class, Insecta. 4. Subclass, Pterygota *5. Order, Hymenoptera. 6. Suborder, Apocrita. 7. Superfamily, Apoidea *8. Family, Apidae. 9. Subfamily, Apinae. 10. Genus, Apis. 11. Species, Indica. Obligate, while others optional. Subspecies is the geographic variety or race. Species is the natural reproductive unit among animals. It is a group of individuals having similar structure, development and behavior which interbreed to produce viable offsprings. Biological species concept says that a species is a natural, interbreeding population which is reproductively isolated from the individuals of other species. Functions of species. I. Reproductive community. The individuals of the species are able to recognize each other as potential mate and seek each other for the purpose of reproduction. E. An ecological unit. Irrespective of the number of individuals, they act as a unit and interact with the individuals of other species with which they share the habitat. E. A genetic unit. It consists of a large number of intercommunicating gene pool in which the individual is a temporary reservoir holding a portion of genes for a short period. Body text young one by parthenogenesis. I. Larval poidogenesis, e.g. gall midges. 26. Nomenclature and identification. A name is required for identification of any organism. Generally, two types of names are used: I. Common name, E. Scientific name. I. Common name. I. They are inaccurate because it varies from region to region and country to country and there is no uniformity followed in naming the organisms. E.g. Locust is a bug referring cicada in European countries, and normally locusts also refer to short-horned grasshoppers living in groups. E. Common name is not available for all organisms. E.g. Squash bugs present in cucurbitaceous plants are represented by many species, but no common name is available for each species. It is available only for a large group like order and family. E. Same common name is used for insects of different orders. E.g. Flies. A true fly has only 2 wings, whereas other insects like mayfly, dragonfly, etc., are also mentioned as flies. IV. Homonym. Same name is used for describing 2 different types of insects. E.g. Bollworm is a common term used for more than 5 species of boll-feeding insects. V. Synonym. More than one name denoting a single insect. E.g. Grampod borer. American bollworm denotes Helicoverpa armigera 2. Scientific name. The system of naming organisms using 2 words is called binomial nomenclature, trinomial nomenclature if 3 words are used. This system of naming gives accurate information. It is universal and is accepted in all parts of the world. The rule regarding the naming of organisms is contained in International Code for Zoological Nomenclature. Normally there are 2 names. The first name is the generic name and the second name is the species name. The names that follow the generic name are called trivial names. The trivial names may be either species or a subspecies name. E.g. Head louse, Pediculus humanus captus. Body louse, Pediculus humanus corporis. The first letter of the generic name is in capital and the first letter of species and subspecies are in small letter. All the words are Latinized and written in italics, or it should be written and underlined separately. The authority name is written after the species name. It starts with capital letter. The author name is put in bracket if the taxa has been reclassified and placed in another group. E.g. The author name is put in bracket if the taxa has been reclassified and placed in another group. E.g. Moringa fruit fly Guidona distigma, mygon. Identification. All insects present in the world are not yet identified and described. About 25% of the insect species are unidentified. An already described species produce new race which requires further identification. Identification helps to understand about the organism and to take proper control measure. Insects can be identified through an expert, by comparing the available collections, using photographs and pictures, and by using taxonomic keys. Key is a tabular statement presenting alternatives, describing about the features of an organism. Most of the keys are dichotomous, i.e., always dividing into 2, or they are always in the form of couplets and give a clear-cut alternative. Keys can be constructed based on single character, monothetic key, or many characters, polythetic key. Polythetic key is more advantageous. Monothetic key has 3 disadvantages. I. The organism may be an exception for a particular character. E. Chances of erring is more. E. If the particular body part is broken on which the key is made, then the key cannot be used. Key can be classified based on evolutionary principles also as phylogenetic key and arbitrary key. I. Fibrous cocoon fibers, red plam weevil. Frassy cocoon. Fibrous cocoon fibers, red plant. Nomenclature and identification. Taxonomy is the science of classification. It can be defined as placing biological organisms or forms in order. Simpson, 1961, has defined taxonomy as the theoretical study of classification including basis, principles, procedures and rules. Taxonomy includes nomenclature and classification. Systematics, the science of study of kind and diversity of organisms and any or all relations among them. Systematics includes taxonomy and evolution. Stages of taxonomy. The taxonomy of any group passes through several stages. I alpha taxonomy alpha, it is concerned with naming and characterization of species. E beta taxonomy beta, concerned with classification. E gamma taxonomy gamma, concerned with evolutionary relations and phylogeny. Basis for classification. Classification is the ordering of a large group of organisms based on certain characters into small groups. Classification is mainly based on evolutionary relationship and not based on superficial resemblance. Points considered while classifying are I. External structure, E. Internal characters, E. Developmental history, IV. Physiological data and V. Cytogenetic data. The biological system of classification is called hierarchical concept of classification. This was introduced by Carl von Linnaeus, 1758. A large group of organism is successively subdivided into small group. These groups are called taxa, taxon singular. Each group is at a particular level in this system. This level is called the rank. Groups of the same rank are grouped together and that constitutes the taxonomic category. E.g., class. Certain taxonomic categories are obligate, while others are optional. For describing and classifying any organism, the basic taxonomic category is species. The lowermost category for classifying an organism is subspecies. Systematic position of Indian honeybee. 1. Kingdom, Animalia. 2. Phylum, Arthropoda. 3. Class, Insecta. 4. Subclass, Pterygota *5. Order, Hymenoptera. 6. Suborder, Apocrita. 7. Superfamily, Apoidea *8. Family, Apidae. 9. Subfamily, Apinae. 10. Genus, Apis. 11. Species, Indica. Obligate, while others optional. Subspecies is the geographic variety or race. Species is the natural reproductive unit among animals. It is a group of individuals having similar structure, development and behavior which interbreed to produce viable offsprings. Biological species concept says that a species is a natural, interbreeding population which is reproductively isolated from the individuals of other species. Functions of species. I. Reproductive community. The individuals of the species are able to recognize each other as potential mate and seek each other for the purpose of reproduction. E. An ecological unit. Irrespective of the number of individuals, they act as a unit and interact with the individuals of other species with which they share the habitat. E. A genetic unit. It consists of a large number of intercommunicating gene pool in which the individual is a temporary reservoir holding a portion of genes for a short period. Body text young one by parthenogenesis. I. Larval polydogenesis, e.g. gall midges. 26. Nomenclature and identification. A name is required for identification of any organism. Generally, two types of names are used: I. Common name, E. Scientific name. I. Common name. I. They are inaccurate because it varies from region to region and country to country, and there is no uniformity followed in naming the organisms. E.g., locust is a bug referring cicada in European countries, and normally locusts also refer to short-horned grasshoppers living in groups. E. Common name is not available for all organisms. E.g., squash bugs present in cucurbitaceous plants are represented by many species, but no common name is available for each species. It is available only for a large group like order and family. E. Same common name is used for insects of different orders. E.g., flies. A true fly has only 2 wings, whereas other insects like mayfly, dragonfly, etc., are also mentioned as flies. IV. Homonym. Same name is used for describing 2 different types of insects. E.g., bollworm is a common term used for more than 5 species of boll-feeding insects. V. Synonym. More than one name denoting a single insect. E.g., grampod borer, American bollworm denotes Helicoverpa armigera 2. Scientific name. The system of naming organisms using 2 words is called binomial nomenclature, trinomial nomenclature if 3 words are used. This system of naming gives accurate information. It is universal and is accepted in all parts of the world. The rule regarding the naming of organisms is contained in International Code for Zoological Nomenclature. Normally there are 2 names. The first name is the generic name and the second name is the species name. The names that follow the generic name are called trivial names. The trivial names may be either species or a subspecies name. E.g., head louse, Pediculus humanus captus. Body louse, Pediculus humanus corporis. The first letter of the generic name is in capital and the first letter of species and subspecies are in small letter. All the words are Latinized and written in italics, or it should be written and underlined separately. The authority name is written after the species name. It starts with capital letter. The author name is put in bracket if the taxa has been reclassified and placed in another group. E.g., moringa fruit fly Gitona distigma, mygon. Identification. All insects present in the world are not yet identified and described. About 25% of the insect species are unidentified. An already described species produce new race which requires further identification. Identification helps to understand about the organism and to take proper control measure. Insects can be identified through an expert, by comparing the available collections, using photographs and pictures, and by using taxonomic keys. Key is a tabular statement presenting alternatives, describing about the features of an organism. Most of the keys are dichotomous, i.e., always dividing into 2, or they are always in the form of couplets and give a clear-cut alternative. Keys can be constructed based on single character, monothetic key, or many characters, polythetic key. Polythetic key is more advantageous. Monothetic key has 3 disadvantages. The organism may be an exception for a particular character. Chances of erring is more. If the particular body part is broken on which the key is made, then the key cannot be used. Key can be classified based on evolutionary principles also as phylogenetic key and arbitrary key. Phylogenetic key. The key is based on the evolutionary relationship. The group appears only once in the phylogenetic key. Arbitrary key. The taxa or group appears at several places in the key. It has more advantages. Chapter 18. Classification of insects in different groups. Insects are the 6-legged flying arthropods coming under the phylum Arthropoda and class Insecta. The widely accepted classification of insects was proposed the eminent insect taxonomist A. Diems. Characters of class Insecta. 1. Body is divided into 3 regions. 2. In head a pair of antennae and a pair of compound eyes are usually present. 3. Thorax is the center of locomotion with 3 pairs of 5-jointed legs and 2 pairs of wings. 4. Excretion is mainly through Malpighian tubules. 5. Tracheal system of respiration well developed. 6. Brain is divided into protocerebrum, detocerebrum and tritocerebrum. Entognathes hexapods. 1. Order, Proteroproterans. 2. Order, Columbola springtails. 3. Order, Dipleura Apterygote hexapods. 4. Microcoryphea bristletail. 5. Thysanura, silverfish. Pterygote hexapods. 6. Ephemeroptera, mayflies. 7. Odonata, dragonflies and damselflies. 8. Orthoptera, grasshoppers, crickets and katydids. 9. Phasmatodea, walking sticks and leaf insects. 10. Gryloblatodea, rock crawlers. 11. Mantophasmatodea, gladiators. 12. Dermaptera, earwigs. 13. Plecoptera, stoneflies. 14. Embiodina, web spinners. 15. Zoroptera, angel insects. 16. Isoptera, termites. 17. Mantodea, mantid. Chapter 18. Classification of insects in different groups. Insects are the 6-legged flying arthropods coming under the phylum Arthropoda and class Insecta. The widely accepted classification of insects was proposed the eminent insect taxonomist A. Diems. Characters of class Insecta. 1. Body is divided into 3 regions. 2. In head a pair of antennae and a pair of compound eyes are usually present. 3. Thorax is the center of locomotion with 3 pairs of 5-jointed legs and 2 pairs of wings. 4. Excretion is mainly through Malpighian tubules. 5. Tracheal system of respiration well developed. 6. Brain is divided into protocerebrum, detocerebrum and tritocerebrum. Entognathes hexapods. 1. Order, Proteroproterans. 2. Order, Columbola springtails. 3. Order, Dipleura Apterygote hexapods. 4. Microcoryphea bristletail. 5. Thysanura, silverfish. Pterygote hexapods. 6. Ephemeroptera, mayflies. 7. Odonata, dragonflies and damselflies. 8. Orthoptera, grasshoppers, crickets and katydids. 9. Phasmatodea, walking sticks and leaf insects. 10. Gryloblatodea, rock crawlers. 11. Mantophasmatodea, gladiators. 12. Dermaptera, earwigs. 13. Plecoptera, stoneflies. 14. Embiodina, web spinners. 15. Zoroptera, angel insects. 16. Isoptera, termites. 17. Mantodea, mantids. 18. Blatodea, cockroaches. 19. Hemiptera, true bugs, cicadas, hoppers, psyllids, whiteflies, aphids and scale insects. 20. Thysanoptera, thrips. 21. Socoptera, sosids. 22. Phyraptera, lice. 23. Coleoptera, beetles and weevils. 24. Neuroptera, alderflies, dobsonflies, fishflies, snakeflies, lacewings, antlions, and owlflies. 25. Hymenoptera, sawflies, parasitic wasps, ants, wasps and bees. 26. Trichoptera, caddisflies. 27. Lepidoptera, butterflies and moths. 28. Siphonoptera, fleas. 29. Mecoptera, scorpionflies and hangingflies. 30. Strepsiptera, twisted-wing parasites. 31. Diptera, flies. The class Insecta has 2 subclasses viz. Apterygoda and Pterygoda. Apterygoda Pterygoda. 1. Primarily wingless, evolved winged or secondarily wingless from wingless ancestors. Evolved from winged ancestors e.g. flea, headlouse, bedbug. 2. Metamorphosis is totally present. Absent or slight. 3. Mandibular articulation in dicondylic i.e., double. Head is monocondylic i.e. single. 4. Pleural sulcus in thorax is absent. Present. 5. Progenital abdominal absent. Dages present. The subclass Apterygoda has 4 orders namely. 1. Thysanura silverfish, thysan fringed, uratail. 2. Columbola springtail or snowflea, call glue, embole peg. 3. Proteroproterans or telsintail pro first, uratail 4. Diplura diplurans or Japygids D2. Uratail, the subclass Pterygoda has two divisions, namely Exopterygoda and Endopterygoda based on the wing development. Character Exopterygoda Endopterygoda. 1. Wing development external internal. 2. Metamorphosis incomplete complete holometabola hemimetabola. Or gradual. Parametabola. 3. Pupal stage absent present. 4. Immature stage naiad or nymph larva. 5. No. Of orders 9-16. The class Insecta is divided into 29 orders, 4 in Apterygoda and 25 in Pterygoda. EXOPTERYGOTA groups. 01. Ephemeroptera mayflies I Paleopteran orders 1, 2. 02. Odonata dragonfly, damselfly. 03. Plecoptera stonefly 2. Orthopteroid orders 3 to 11. 04. Gerlobolatodia rock crawlers. 05. Orthoptera grasshopper, locust, cricket, mole cricket. 06. Phasmata stick insect, leaf insect. 07. Dermaptera earwigs 08. Embioptera web spinners, embids. 09. Dictyoptera cockroach, praying mantis. 10. Isoptera termites. 11. Zoraptera zorapterans. 12. Socoptera book lice 3. Hemipteroid orders 12 to 16. 13. Melophaga bird lice. 14. Siphonculata head and body louse. 15. Hemiptera bugs 16. Thyssanoptera thrips. ENDOPTERYGOTA. 01. Neuropterontilians, aphid lion, owl flies, mantispid flies. 02. Macoptera scorpion flies. 03. Lepidoptera butterflies and moths. 04. Trichoptera caddisfly. 05. Diptera true fly. Group 4. Panorpoid complex 1 to 6. 06. Siphonoptera fleas. 07. Hymenoptera bees, wasps, ants. 08. Coleoptera beetles and weevils. 09. Strepsiptera stylopids. Paleopteran insects cannot flex the wings over the abdomen, i.e., wing flexing mechanism is absent. The characters of insects belonging to Orthopteroid group include mandibulate mouth parts, anal area of hind wing well developed, abdomen is always with cerci, many numbers of Malpighian tubules and the ganglia of the ventral nerve cord are not fused. Hemipteroid insects have the haustellate mouth parts and mainly they feed on liquid food like plant sap. Panorpoid insects are holometabolous and they have larval and pupal stage. Chapter 19. Study of insect orders. Protura, Collembola, Diplura, Microcoryphea. 1. Order. Protura proturans. The proturans are minute whitish hexapods, 0.6 to 1.5 mm in length. The head is somewhat conical, and there are no eyes or antennae. The mouth parts do not bite, but are apparently used to scrape off food particles that are then mixed with saliva and sucked into the mouth. The first pair of legs is principally sensory in function and is carried in an elevated position like antennae. The tarsi are one-segmented. Styli are present. The proturans are minute whitish hexapods, 0.6 to 1.5 mm in length. The head is somewhat conical, and there are no eyes or antennae. The mouth parts do not bite, but are apparently used to scrape off food particles that are then mixed with saliva and sucked into the mouth. The first pair of legs is principally sensory in function and is carried in an elevated position like antennae. The tarsi are one-segmented. Styli are present on the first 3 abdominal segments. On hatching from the egg, the proturan abdomen consists of 9 segments. At each of the next 3 molts, segments are added anterior to the apical portion, the telson, so that the adult abdomen appears to have 12 segments, 11 metameric segments and the apical telson. These hexapods live in the moist soil or humus, in leaf mold, under bark, and in decomposing logs. They feed on decomposing organic matter and fungal spores. They are found worldwide and approximately 200 species are known at present. Important identification characteristics. Minute insects, with entignathous piercing mouth parts B, antennae and eyes are absent. C. Abdomen 11 segmented with a terminal telson, first 3 segments with a pair of small appendages called styli D, forelegs sensory and held above the head, like antennae. E. Metamorphosis slight, chiefly evident as an increase in number of abdominal segments following each molt, anamorphosis, e.g. Eosentomen indicus. 2. Order, Collembola. Synonyms, Oligentoma, Oligontomata. Etymology, caul glue, embol wedge or peg. Common names, springtail, snow flea. Characters, they are minute insects. Body is globose or tubular. Compound eyes are absent. 1 to several pairs of lateral ocelli form an eye patch. Antenna is 4-segmented. Mouth parts are entignathous biting type and found within a pouch. Tibia is fused with tarsus to form TBO tarsus. They are primarily wingless. Abdomen is 6-segmented with their medially situated progenital appendages. Ventral tube or coliphore or glue peg. It is a bilobed adhesive organ found on the first abdominal sternite. It is believed to be associated with respiration, adhesion and water absorption. Hemula or tenaculum or retinaculum. It is present on the third abdominal sternite. It consists of a fused basal piece, corpus and free distal part called rami. It holds the furcula. Furcula or springing organ. It consists of a basal manubrium, paired dens and distal claws called mucro. It is held under tension beneath the abdomen by retinaculum when at rest. Malpighian tubules, tracheal system and metamorphosis usually absent. Importance. Sminthurus viridis is a pest on alfalfa. It can be collected from moist places in soil. They are also found in mushroom houses as a pest. 3. Order, Diplura diplurans. The diplurans appear somewhat similar to the silverfish and bristletails, but they lack a median caudal filament and thus have only 2 caudal filaments or appendages. The body is usually not covered with scales. Compound eyes and ocelli are absent. The tarsi are one-segmented, and the mouth parts are mandibulate and withdrawn into the head. The antennae are long and multisegmented. Styli are present on abdominal segments 1 to 7 or 2 to 7. These hexapods are small, generally less than 7 mm in length, and usually pale in color. They are found in damp places in the soil, under bark, under stones or logs, in rotting wood, in caves, and in similar moist situations. Identification characteristics. A. Small to large, narrow-bodied Entognathus insects. B. Antennae maniliform, with intrinsic musculature. C. Compound eyes and ocelli absent. D. Abdomen with 10 segments, ending in a pair of cerci. E. Larval development epimorphic. E.g. Campodia staphylinus. E.g. Smintheris viridis. Class, Insecta. Subclass, Apterygota. 4. Order, Microcoryphea equals Archaeognatha, bristle tails. The Microcoryphea resemble the silverfish in the order Thysanura. However, they are more cylindrical, with the thorax somewhat arched. The compound eyes are large and contiguous. Ocelli are always present. Each mandible has a single point of articulation with the head capsule. The tarsi are 3-segmented, and the middle and hind coxae usually bear styli. These styli are sometimes lacking on the middle coxae, or they may be completely absent. The abdomen bears a pair of styli on segments 2 to 9, and segments 2 to 7 each bear 3 ventral sclerites, the coxopodites, and a median sternum. The sternum is sometimes much reduced. Segments 1 to 7 usually bear 1 or 2 pairs of eversible vesicles. These insects live in grassy or wooded areas under leaves, under bark, in dead wood, under stones, under rocks and cliffs, and in similar situations. Most are nocturnal, and their eyes glow at night when illuminated with a flashlight. The largest members of the order are about 15 mm in length. The Microcoryphea are quite active and jump when disturbed, sometimes as far as 25 to 30 cm. The eversible vesicles on the abdomen function as water-absorbing organs. Before these insects molt, they cement themselves to the substrate. The cement appears to be fecal material. If the cement fails, or if the substrate, such as sand, is not firm, they are unable to molt, and die. The bodies of the insects are covered with scales, which sometimes form distinctive patterns. The scales are often lost during the collecting process or when the insects are preserved in fluid. The jumping bristletails feed chiefly on algae, but feed also on lichens, mosses, decaying fruits, and similar materials. Identification characteristics. A. Fusiform, subcylindrical, apterygotes with ability to jump. B. Body-bearing pigmented scales. C. Compound eyes large, contiguous, ocelli present. D. Mandibles with single articulation, maxillary palpi 7 to 9 segmented. E. Thorax strongly arched, terga extending over pleura. F. Styli often present on mid and hind coxae, tarsi 3 segmented. G. Abdominal segments 2 to 9 with ventral styli, 1 to 7 with eversible vesicles. E.g. Graphotarsus pseudindicus. Chapter 20. Study of insect orders. Thysanura, Ephemeroptera, Odonata. 8. Order, Thysanura. Synonyms, Ectognatha, ectotrophy. Etymology, Thysan fringe, ura, tail. Common names, silverfish, fire brat, bristle tail. Characters, body is elongate and flattened. Body is glistening and clothed with scales. Compound eyes are present or absent. Antennae is long, filiform and multisegmented. Mouth parts are ecotognathous, biting type. They are primarily wingless insects. Abdomen is 11-segmented. Varying number of bilateral styli are present on abdominal sternites. Styli are bellive to be reduced abdominal legs. Female has elongate jointed ovipositor. Abdomen at its tip carries a pair of elongate many-segmented cerci and a median caudal filament. Insemination is indirect. Metamorphosis is absent. Molting continues even after attaining sexual maturity. Importance. It is often a pest in home and libraries. Ctenalepisma sp. is the common household silverfish. It feeds and destroys paper, book bindings and starched clothing. It can be collected from amongst old books, behind calendar, photo frames, etc. Class, Insecta. Subclass, Pterygota 9. Order, Ephemeroptera. Synonyms, Ephemerida, Plectoptera. Etymology, ephemeral living for a day, ptera wing. Common names, mayflies, shadflies, dayflies. Characters, small to medium-sized soft-bodied insects. Compound eyes are large. There are 3 ocelli. Antenna is short and setaceous. Mouth parts in adults are atrophied. Forewings are large and triangular. Hind wings are small and absent in some species. Numerous cross and intercalary veins are present. Wings are held vertically over the abdomen. Wing flexing mechanism is absent. Abdomen is slender with a pair of long cerci. Median caudal filament may be present or absent. Metamorphosis is incomplete with 3 stages viz. Egg, naiad and adult. Naiad is aquatic with biting mouthparts. It breathes through bilateral abdominal gills. At tip of the abdomen a pair of long cerci and a median caudal filament are usually present. Immediately after the adult emergence body of the insect is covered with closely set fine hairs called pellicle and this stage is called as subimago. It is dull in color with opaque wings and legs and cerci are not well developed. In imago wings are transparent. Legs and cerci are well developed. Body is shiny and not covered with pellicle. Adults are found near lakes and ponds and are also attracted by light. Importance. Naiads are important fish food. Adults are short-lived and hence the name dayfly. When they emerge in large numbers they pose nuisance problem. 10. Order, Odonata. Etymology, odin tooth. Common names, dragonflies and damselflies. Characters, medium to large-sized insects. They are attractively colored. Head is globular and constricted behind into a petiolate neck. Compound eyes are large. 3 ocelli are present. Mouth parts are adapted for biting. Mandibles are strongly toothed. Lacenia and Galea are fused to form mala, which is also toothed. Wings are either equal or subequal. Membranous. Venation is network-like with many cross veins. Wings have a dark pterostigma towards the costal apex. Subcosta ends in nodus. Wing flexing mechanism is absent. Legs are anteroventrally placed. They are suited for grasping, holding and conveying the prey to the mouth. Spinous femora and tibia are useful for holding the prey. Forward shift of leg attachments allow easy transfer of prey items to mouth in flight. Legs are held in such a way that a basket is formed into which the food is scooped. Abdomen is long and slender. In male gonopore is present on 9th abdominal segment. But the functional copulatory organ is present on the 2nd abdominal sternite. Before mating, sperms are transferred to the functional penis. Circus is one-segmented. Metamorphosis is incomplete with 3 life stages. The naiad is aquatic. Labium is greatly elongated, jointed and bears 2 hooks at apex. It is called mask. It is useful to capture the prey. Importance. Adults are aerial predators. They are able to catch, hold and devour the prey in flight. Naiads are aquatic predators. Dragonflies and damselflies can be collected with an aerial net near streams and ponds, especially on a sunny day. Naiads can be collected from shallow freshwater ponds and rice fields. Classification. There are 2 suborders. Dragonflies are classified under Anisoptera and damselflies are grouped under Zygoptera. Chapter 21. Study of insect orders, Orthoptera. 11. Order, Orthoptera. Synonyms: Saltatoria, Saltatoptera. Etymology: Ortnostrate, ptera wings. Common names: grasshoppers, locust, katydid, cricket, mole cricket. Characters: they are medium to large-sized insects. Antenna is filiform. Mouth parts are mandibulate. Prothorax is large. Pronotum is curved, ventrally thickened and known as tegmina. They are capable of bending without breaking. Hindwings are membranous with large anal area. They are folded by longitudinal pleats between veins and kept beneath the tegmina. Cerci are short and unsegmented. Ovipositor is well developed in female. Metamorphosis is gradual. In many orthopterans, the newly hatched first instar nymphs are covered by loose cuticle and are called pronymphs. Wing pads of nymphs undergo reversal during development. Specialized stridulatory, sound-producing, and auditory hearing organs are present. Classification. This order is subdivided into 2 suborders, viz., Calyphera and Encephera. Calyphera encephera. 1. Antenna is short with less than antenna is long with more than 30 segments. 2. Tympanum is found on the lateral. Tympanum is found on the side of the first abdominal, the foratibia segment. 3. Vision and hearing. Acute tactile response is well developed. 4. Mandibles are specialized for feed on dicot plants consuming monocot foliage. 5. Diurnal nocturnal. 6. Rely on jumping to escape from. Rely on crypsis predators. 7. Eggs are laid in groups in soil. Eggs are singly inserted inside shallow burrows. Into plant tissue or soil. I. Suborder, Calyphera. Family, Acrididae, short-horned grasshoppers and locusts. 2. Suborder, Encephera. Families, 1. Tedygonidae, long-horned grasshoppers, katydids and bush crickets. 2. Grillidae, crickets. 3. Grillotalpidae, mole crickets. Chapter 22. Study of insect orders, Phasmida, Grilloblatodia. 12. Order, Phasmida. Synonyms: Phasmodia, Phasmatodia. Etymology: Phasma and apparition. Common names: stick insects, leaf insects. Characters: body is stick-like or leaf-like. Head is prognathous. Mouth parts are chewing type. Prothorax is short. Meso and metathorax are long. Metathorax is closely associated with the first abdominal segment. Legs are widely separated. They are long and slender, resembling twigs in stick insect. Tibia and femur shows limellate expansion in leaf insects. A line of weakness is found between the trochanter and rest of the leg. The legs get broken easily at this region and such legs get regenerated subsequently. Tarsus is 5-segmented. Wings may be present or absent. Forewings when present are small and modified into tegmina. In leaf insects, the wing venation mimics leaf venation. Cerci are short and unsegmented. They show protective resemblance. They are herbivorous. Classification. There are 2 families. Stick insects are grouped under Phasmidae and leaf insects are classified under Phyllidae. 13. Order, Grilloblatodia, rock crawlers. The first member of this group was not discovered until 1914, when Walker described Grilloblata campodiformis from Banff, Alberta. Rock crawlers are slender, elongate, wingless insects, usually about 15 to 30 m in length. The body is pale in color and finely pubescent. The eyes are small or absent, and finally are no ocelli. The antennae are long and filiform, consisting of 23 to 45 segments. The cerci are long, with either 5 or 8 segments, and the sword-shaped ovipositor of the female is nearly as long as the cerci. There are only 25 species and 4 genera of living rock crawlers in the world. Rock crawlers live in cold places such as the talus slopes at the edges of glaciers and in ice caves, often at high elevations. They are mainly nocturnal, and their principal food appears to be dead insects and other organic matter found on the snow and ice fields. They are soft-bodied, and probably best preserved in alcohol. Identification characteristics. A. Slender, elongated wingless insects, 15-30 mm. B. Body is pale and finely pubescent. C. Eyes are small or absent and no ocelli. D. Antennae long filiform. E. Cerci are long and the sword-shaped ovipositor, e.g. Grilloblata sp. Chapter 23. Study of insect orders, Mantophasmotodia, Dermaptera. 14. Order, Mantophasmotodia, gladiators. The Mantophasmotodia is the newest addition to the cast of insect orders. The genus Raptophasma was first described in 2001 for fossil specimens preserved in Baltic amber, approximately 30 million years old. Specimens of similar species were later discovered in collections from Namibia and Tanzania in Africa, and a new order was proposed for them. Mantophasmatodia have since been posed for them. Mantophasmatodia have since been found in a number of locations in South Africa in Cape Faunal Zone, an area well known for species richness and endemism. The order contains only single family, Mantophasmatidae, and 3 genera, the fossil Raptophasma, 1 described species, and the living genera Mantophasma, 2 species, and Predatophasma, 1 species. Researchers think there are at least 3 new species in South Africa. Mantophasmatodia are rather small, generally 2 to 3 cm in length, and both sexes are singles. They have chewing mouthparts, the head is hypognathous, and the antennae are long and filiform. The tarsi are 5-segmented. They have simple metamorphosis. According to Van Noort 2003, Mantophasmatodia superficially resemble immature mantids, but the 4 legs are not modified for prey capture. Nevertheless, these are predatory insects. Adults are rather short-lived, surviving for only a few weeks. In the original description of the order, it was unclear which orders of insects were the closest relatives of Mantophasmatodia, but Groloblatodia and Phasmatodia were suggested as possibilities. Unfortunately, no phylogenetic analysis preceded the description. It is unclear, at this early point, whether the recognition of this group as an order will meet with general acceptance or if it will eventually be subsumed within Groloblatodia or another group. Identification characteristics. A. Small, generally 2 to 3 cm in length. B. Both sexes are wingless. C. Have chewing and biting types of mouth part. D. Head is hypognathous. E. Antennae are long filiform. F. Tarsi are 5-segmented. G. The newest order added to the class Insecta, e.g. Predatophasma morazi. 15. Order, Dermaptera. Synonyms, Euplexoptera, Eupolcoptera. Etymology, derma skin, ptera wing. Common names, earwigs. Characters, they are generally elongate insects. Head is with a distinct Y-shaped epicranial suture. They have chewing mouthparts. Prothorax is large, well-developed and mobile. Meso and metathorax are fused with the first abdominal segment. Forewings are short, leathery and veinless. Both the wings meet along a mid-dorsal line. They are called tegmina or elytra. They are protective in function and are not used for flight. Hindwings are large, membranous, semicircular and ear-like. The anal area of the wings is large with a number of branches of anal veins which are radially arranged. They are folded fan-like, longitudinally and twice transversely and kept beneath the forewings at rest. Wings do not cover the abdomen fully. Cerci are found at the end of the abdomen. They are unsegmented enlarged, highly sclerotized and forceps-like. They are large and bowed in male and nearly straight in female. They are useful in defense, folding and unfolding of wings, prey capture and copulation. Parental care is shown by female earwigs. It literally, roost on the eggs until hatching occurs and also cares for the nymphs. Importance. Euborrelia annulipe bores into groundnut pods and feeds on the kernel. Chapter 24. Study of insect orders. Plecoptera, Ambioptera. 16. Order, Plecoptera, stoneflies. Stoneflies are mostly medium-sized or small, somewhat flattened, soft-bodied, rather drab-colored insects found near streams or rocky lake shore. They are generally poor flyers and are seldom found far from water. Most species have 4 membranous wings. The front wings are elongate and rather narrow and usually have a series of crossveins between M and Q1 and between Q1 and Q2. The hind wings are slightly shorter than the front wings and usually have a well-developed anal lobe that is folded fanwise when the wings are at rest. A few species of stoneflies have reduced wings or lack wings, usually in the male. Stoneflies at rest hold the wings flat over the abdomen. The antennae are long, slender, and many-segmented. The tarsi are 3-segmented. Cerci are present and may be long or short. The mouthparts are of the chewing type, although in many adults, which do not feed, they are somewhat reduced. The stoneflies undergo simple metamorphosis, and the nymphal stages of development are aquatic. Stoneflies nymphs are somewhat elongate, flattened insects with long antennae and long cerci, and often with branched gills on the thorax and about the bases of the legs. They are very similar to mayfly nymph but lack a median caudal filament, that is, they have only 2 tails, whereas mayfly nymphs nearly always have 3. Stonefly nymphs have 2 tarsal claws and mayfly nymphs have only 1, and the gills are different. Mayfly nymphs have leaf-like gills along the sides of the abdomen, whereas stonefly gills are always finger-like, either simple or branched, and only occur ventrally. Stonefly nymphs are often found under stones in streams or along lake shores, hence the common name of these insects, but may occasionally be found anywhere in a stream where food is available. A few species live in underground water, and their nymphs sometimes appear in wells or other drinking water supplies. Some species are plant feeders in the nymphal stage, and others are predaceous or omnivorous. Some species of stoneflies emerge, feed, and mate during the fall and winter months. The nymphs of these species are generally plant feeders, and the adults feed chiefly on blue-green algae and are diurnal in feeding habits. The species that emerge during the summer vary in nymphal feeding habits. Many do not feed as adults. In many species of stoneflies, the sexes get together in response to acoustic signals. The males drum by tapping the tip of the abdomen on the substrate. Virgin females respond to this drumming, and answer with a drumming of their own either during or immediately after the male drumming. The males drum throughout their adult life, and the signals are species-specific. Identification characteristics. A. Soft-bodied insects with long setaceous antennae. B. Mouth parts weak, biting type, mandibles normal or vestigial, ligula 4-lobed. C. Wings membranous, held flat over the body at rest, hind pair usually larger, with well-developed anal lobes. D. Tarsi 3-segmented, cerci long. E. Naiads respire through filamentous tracheal gills e.g. Hemera punjabensis. 17. Order, Ambioptera. Synonyms: Ambiodia, Ambiodina. Etymology: Ambia lively, Terra wings. Common names: Ambeads, web spinners.